Howdy, my name is Jazlyn Gonzalez, and I am part of group number 6 with Andres Garza. Today we will be going over PCR and gel electrophoresis. Polymerase chain reaction, or PCR, is a technique used to make many copies of a specific piece of DNA, which makes it easier to detect and study. It works by repeatedly heating and cooling the DNA in the presence of primers, nucleotides, and a special heat-stable DNA polymerase. PCR is important as it allows scientists to test for the presence of certain genes or organisms, look for mutations, and analyze DNA from small or degraded samples. Gel electrophoresis is a method used to separate pieces of DNA based on their size. DNA samples are placed into wells in a gel, and an electric current is run through it. Because DNA is negatively charged, it moves towards the positive end, and smaller fragments travel farther through the gel than the larger ones. After the run, the DNA appears as bands that can be visualized under special light or stain. Gel electrophoresis is important because it lets us see the results of our PCR. Now for our materials. For media, we used master mix, primer mix, restriction enzyme, gel matrix, buffer solution, and for our DNA samples, we had our non-pathogenic control, our pathogenic control, lettuce, and tomato DNA. For equipment, we used 8 0.2 milliliter PCR tubes, a microcentrifuge, thermocycler, a micropipettes and the micropipette tips, a gel casting tray, a comb, and an electrophoresis chamber. Now for our procedure. We first began by setting up our PCR samples. Here, 4 0.2 milliliter PCR tubes were labeled with 1, 2, 3, and 4. Using a micropipette, 15 microliters of master mix were added to each tube, followed by 10 microliters of primer mix also added to each tube. Respectively to each tube, 5 microliters of DNA sample, being our non-pathogenic control, pathogenic control, lettuce, and tomato, were added to each of the labeled tubes, totaling to a total volume of 30 microliters each tube. Each tube was then flicked as well as put into the microcentrifuge for a brief spin to ensure that there was no liquid on the sides of the tubes. We then ran PCR samples. The thermocycler was programmed with the following parameters by the lab techs. The PCR took approximately 45 minutes. I am not sure whether the lab techs put it in the fridge or left it at room temperature, but what I do know is that the PCR product is stable at room temperature for several days, which then needs to be moved to a freezer for longer term storage. Then we did our restriction digestion. Here, 4 0.2 milliliter PCR tubes were labeled with R1, R2, R3, and R4. Using a micropipette, 15 microliters of PCR product from tube 1 was transferred to tube R1. Using a new tip each time, we repeated this for the other 3 tubes. 2 microliters of diluted restriction enzyme was then added to each of our samples labeled R1, R2, R3, and R4. Each tube was flicked as well as put into the microcentrifuge for a brief spin to ensure that there was no liquid on the sides of the tubes. These tubes were incubated at 37 degrees Celsius overnight. Now for agarose gel preparation and gel electrophoresis. 50 milliliters of TBE buffer and 0.75 grams of agarose powder were added into a flask and swirled. It was then microwaved for about 2.5 minutes, ensuring it was clear. After allowing it to cool, 5 microliters of cybergreen were added to the flask and gently swirled before being poured into the gel tray that was previously prepared with the comb in its correct position. Any bubbles were popped using pipette tips, and the gel was allowed to solidify before it was being wrapped in plastic wrap, placed into a Ziploc bag, and then wrapped in foil paper until the next class time. The gel was placed into the electrophoresis chamber with the wells being closer to the black electrode. The chamber was filled with TBE buffer, ensuring the gel was submerged. Using a micropipette, our samples were loaded into the following order, changing out tips to prevent contamination. For lane 2, we used 10 microliters of our fast DNA ladder. Lane 3 was 10 microliters of sample R1. Lane 4 was 10 microliters of sample R2, Lane 5 was 10 microliters of sample R3, and Lane 6 was 10 microliters of sample R4. The gel ran for 30 minutes at 100 volts before being moved to a transilluminator and viewed under blue light.
Moving on to the results and observations, we can see here that on our gel electrophoresis, we have our DNA ladder on the very left-hand side. And then for R1, we have one single band, which indicates that it's non-pathogenic, which means that there is no restriction site. As for R2, we have three distinct bands. This one is pathogenic. It has a pathogenic gene sequence. And for R3, we have one single band, which is also indicating that it is a non-pathogenic with no restriction site. And lastly, for R4, we have two distinct bands, which is a pathogenic gene sequence. And lastly, for our conclusion, overall, this lab successfully demonstrated how PCR and gel electrophoresis work together to identify DNA differences between samples. PCR allowed us to amplify specific DNA regions, and gel electrophoresis helped us visualize those amplified fragments as clear bands on the gel. Based on the banding patterns, we were able to distinguish non-pathogenic samples, which showed a single band, from pathogenic ones that produced multiple bands after restriction digestion. These results confirm that PCR and gel electrophoresis are reliable and essential tools for detecting genetic variation and identifying unknown samples in a laboratory setting. These are also our resources that we use for the lab report. And this concludes our lab report video presentation. Thank you all for listening.